Okay, good afternoon, everybody. This one's going to be a little different, kind of just for fun. So sit back, relax a little bit. You're probably not going to learn anything from this. I just wanted to talk a bit about some of the cool changes that I kind of think might be coming. And let me point out, uh, I do not consider myself an expert in any of these things I'm really going to talk about here. So it's going to be kind of a cursory treatment. If you have any questions at the end, I probably won't do a very good job of answering them. But I just wanted to kind of geek out a little bit and show you some of these things that are coming along. So Microbiology 2040, 16 years from now. This is the kind of presentation that people usually give at the end of their career. I'm not quite there yet, but I'm not going to be around in 2040. So if none of this stuff happens, you can blame whoever it is who takes over for me. I started in this job here, not this job here, but this career in 1991. So this is some of the stuff that's happened in clinical microbiology since that time. Real-time PCR came along, and of course, faster computing has been going on for decades now. We get faster and faster all the time. And then sequencing started up. So all those things have kind of happened since I've been in this business. So now we're in 2024, and all of these things are happening now. All of these things are improving and getting better. And everything that I say here about what might be going on in 2040 is really going to kind of depend on all of these things continuing to get faster and continuing to get more practical. So all these ideas have started. We're just moving a little bit further. But to tell you the truth, if you look back at how technology has changed things in the last 16 years, for example, and the rates just continuing to increase, I kind of think this is where we're probably going to be at that point. And it'll be kind of disappointing if we're not there. So all of these things. All of these things. Okay. Are you still hearing me? Because I have crashed here. Oh, there we go. All of these things are going to come together and just completely blow up our idea of what goes on in a clinical microbiology laboratory. I want to point this out specifically. This is the change that really still needs to occur. When it comes to DNA sequencing, what we can do right now is targeted sequencing, where you do a PCR for specific targets within a bacterial or a fungal genome, and then you sequence those amplicons. And this is something that we already do. Not many laboratories do this in their own laboratory. Not many hospital laboratories do. But you can send these things out pretty routinely now to look for microorganisms in patient specimens. So if you've seen Microgen results or University of Washington Universal PCR results, that's what you're seeing, targeted sequencing. But where we're moving is to more whole genome sequencing, where you don't do those initial PCRs. You just fragment all the DNA that's in that specimen. And then you sequence the fragments and then put them all back together again. So there's not any specificity to it. You're doing bacterial DNA and fungal DNA and viral DNA and human DNA and assembling the whole stuff all back together. Now, we can already do this on isolates. I'll show you a couple of examples how you can use this on bugs that you've already grown. And if you've sent out certain tests, for example, the Carius test, that is one that gets sent out. This is whole genome sequencing that's being done on a simple specimen that doesn't have a whole lot of DNA in it. But here's the deal. It's really not yet practical on most specimen types. That's where we're going to get to. So if I step back and kind of think about what microbiology is like in 1991, when I started here, not here. I shouldn't say that. I've only been here for 15 years. 2009 was when I got here. But in 1991, technology is pretty limited. You know, we streak plates, we look under a microscope. We had incubators. There's a lot of skill involved. And there's still a lot of skill involved in reading bacterial cultures. but the information that you got was fairly limited. Compare that to where we're going in 2040, much expanded technology, a whole lot of new equipment and new abilities to do things that actually require less skill. You don't have to have that technical skill, the technical ability to do things. It's easier to do a lot of this molecular stuff, but you're going to get much more extensive information, logs different in the information. So microbiology has gone from really a technical skill to where it's going to be information management. And we're kind of in that crossroads right now. We're kind of getting to where we are more and more information management. So let's give some examples here on what things are going to be like when you're sitting here listening to next Norton Infectious Disease Institute grand rounds in April of 2040. What you see now, when we do a culture, here's a sputum that came to us, and we do a culture, and we're looking for the clinically significant organism. So we report out something like moderate growth of Club pneumo. That's the bug that we think is the clinically significant organism. And then we say light growth, normal respiratory flora. What we're going to be doing at that point is we're not going to culture at all. We're going to take this specimen, we're going to put it in some sort of an analyzer that will do whole genome sequencing or targeted sequencing. And you'll get a report that looks sort of like this. This is the same specimen. You know, Club pneumo is in there. We actually had more Prevotella, which didn't grow because it's an anaerobe. We don't culture sputums for anaerobes. And then all of this other stuff that would have been lumped together as this normal flora. So you can kind of see where the information management becomes more of an issue of trying to fish out and hear what is clinically significant and what isn't. So what you'll be seeing is a lot more information, a little bit more difficulty in how to handle it. And again, this will be without having to culture it at all. I think it's going to look something like this. You know, here's a big analyzer. We take a sputum specimen, we stick it in some port down here at this end, and sometime later, it might be a couple hours, it might be 12 hours, 24 hours later, something. You're going to get a printout that comes out this side, which is going to tell you all the bugs that it found in that specimen. And we will just keep feeding specimens in over here and getting those results out. You know, people always tell me one of these days microbiology is going to become just like chemistry. And I told them they're crazy. And now I can see that it is going to happen. We are going to become just like the chemistry laboratory. So years down the road, are we going to culture at all? Viral culture, let's take that as an example. That's already gone. You know, not many, very few laboratories do viral culture anymore. We quit maybe like 6 or 7 years ago. It's been replaced by PCR. It's fairly easy to target certain viruses and say we want to do PCRs directly for those viruses. It's faster, it's much higher accuracy, more sensitive than culture is, and it's cheaper than culture. And you don't have to have a whole tissue culture set up and people who are very skilled at doing that. So even right now, viral culture is largely going to be limited to just reference laboratories or public health laboratories. It's something that you send out. We don't send out many viral cultures at all anymore. And to be honest with you, most of the ones that we send out are inappropriate. They're being ordered because it happens to be on the order set, but there's nothing really that they're looking for. So fungal and AFB culture, those are going to be the next ones to go. These are very slow cultures. They're very labor-intensive cultures, and especially fungal culture depends very much upon a skilled technologist, a very clinical laboratory scientist who's got some experience and can look at these things and figure out what they are. You can sequence these fairly simply. Relatively limited number of organisms that you're looking for, at least that are recognized as human pathogens. There's a whole ton of molds out there that are environmental organisms, though. So most of your small labs don't do these at all, and the larger laboratories at some point in the near future are going to completely switch over to sequencing or PCRs for those. Bacterial culture is going to be the hard one to replace. Most of our cultures, probably 70–80% of all our cultures are going to be bacterial cultures, so it's a high volume, tons and tons and tons of different organisms, hundreds, thousands of different organisms that you could be looking for. Lots of different resistance mechanisms that you need to think about. So it's going to be a hard one to get rid of. It'll be the last one to go. But I do think probably at some point in the future, bacterial culture will kind of go the same way that viral culture has gone, where there's only a few large reference laboratories or public health laboratories that do it, and only in those situations where there's something unusual going on. Hospital laboratories might not do it much at all. So, okay, now what you're thinking of, that's great. Okay, you can identify bugs that way. That's fantastic. But what about susceptibility testing? We still need to grow the bugs in order to do susceptibility testing. Let me give you a couple of options here. And first of all, let me tell you that this thing I'm showing you right now, I don't like the idea. I'm sure our infectious disease pharmacists would not like the idea, but I'm just putting it out there to tell you that there is a possibility that at some point we won't culture to grow the organisms. And we'll do this little cultural method right here or something similar to this just to get susceptibility results. These are people that came up with a plan that they have here, where what they do is they culture the whole specimen. You don't split out the bugs. You don't get that clade pneumo out of that sputum and do susceptibility on it. You do susceptibility on the entire specimen. They're looking at it and saying, you got to take into effect interbacterial interactions that affect how bugs are going to behave in the presence of an antibiotic. This is one paper, and then this is the paper where they showed how well it works on cystic fibrosis patients. This is basically the way it works right here. You take the specimen itself, you take a sputum sample or a urine sample or whatever, and you put it in these little wells that have different antibiotics. Incubate it for 4 hours, look at them microscopically to see if you see any bacteria growing. That's the way they do it. To me, I look at it and say, I can't fathom that actually working. I don't like the idea at all. It would take a whole lot to convince me to do it this way. But I wanted to put it out there just to say that perhaps in the future, we may do something like this where we're doing the whole specimen susceptibility and not a single organism susceptibility. But what I think is more likely to happen is that we'll stick with sequencing where we do not have to actually culture the bug at all. We can sequence it. I want to show you this. This is a test that we've actually done. This is a Pseudomonas aeruginosa. We grew the bug. So this was an isolate, okay? And we sent these out for whole genome sequencing and got reports back that look like this. This is a whole lot of information that you can get out of these things from sequencing. It gives us all kind of microbial typing schemes here, tells us there's a plasmid. And here's the susceptibilities. It is predicting the susceptibilities based upon the gene sequences that are present. And you can look at all kinds of known resistance genes, and you can look at all kinds of artificial intelligence algorithms that kind of look at various sequences and kind of figure out what the most likely phenotype is going to be. That might be what we end up doing. This particular isolate, it said that everything was going to be susceptible. All of these drugs would be susceptible. And I can tell you, since we grew this bug, we did susceptibility, and we agree it was totally susceptible. With this one exception, gentamicin had an MIC of 4. We call that resistant. Notice here that gentamicin MIC of 4 should be susceptible, but that's because we have changed our protocol. We never report Pseudomonas aeruginosa as susceptible to gentamicin anymore. It's always resistant to gentamicin. Not suggested that you ever use gentamicin for Pseudomonas. So it worked. So we could do this either on isolates, or we theoretically could do this on a specimen. Not only will you get the whole list of all these organisms that are in that specimen, but you'll actually get predicted susceptibility results for every single one of those organisms. This just shows you how detailed this is. This is that same isolate. These are all the different resistance genes that it looked for, and which ones it found and which ones it didn't. So that is just a pretty impressive list, and not incredibly expensive, not really that difficult to do. So let's go to infection control issues. I wanted to show you this with the whole genome sequencing. When you're looking at epidemiology of an organism, that genome sequence is really like the gold standard. You're looking at the actual genome sequence. So that is, more so than other epidemiological genetic typing methods. That is like the gold standard. That tells you what it is. So here's another project that we worked on. These were isolates that we sent out for whole genome sequencing, a suspected outbreak going on in one of our facilities. And you can look at this. This shows you, based upon that sequence data, how related these organisms were. So here are 4 isolates right here that were extremely closely related. This is group 5. Group 4 has these 2. Here's a big group. Different patients all had pretty much the same isolate of the organism right there. So you can see how easily with sequencing you can kind of put these things together and decide which ones are related. Is Mrs. Smith's E. coli related to Mr. Jones' E. coli? He's in the next room over. If they're not related, then we're not too concerned about that having been spread, right? But if Mrs. Smith's E. coli is exactly the same as Mr. Frazier's E. coli, then we could look for the relationship between those two and see how that spread may have happened, and then stop it. Now, this data that I'm showing you is done on isolated bacteria. Okay, this is whole genome sequencing on isolated bacteria. But if we get to that point, if we get the computing power, and that's really the only thing we're losing, we don't have right now, is the ability to handle that amount of data in a reasonable amount of time on a computer. Once we get to that point, then we could do this directly in specimens. And then we could look at all the organisms that came out of that specimen and, are they related to other organisms that we have isolated? This is being done. There are ways to do this, and there are companies that really will push for you to do this in your hospital. You can do it prospectively. I went to a presentation 6 years ago. 6 years ago, where a hospital in England was showing that what they do is they get a MRSA isolate, they grow it, they get a MRSA isolate, it gets whole genome sequencing. There's another one. So it puts them into these groups right as it comes along. So once you get 2 that match, immediately, bam, that these 2 are related. And then you can keep going and you can keep going, and then you can figure out these possible outbreaks instantly, almost in real time. What this laboratory did is every MRSA that they isolated today got sequenced overnight. And when they came in the next morning, there was the report of the relatedness between them. So immediately, if you have a transmission that took place in your hospital, you know about it within a couple days, and you can do something about it. So it is possible to do that. And that was being done in isolates. But again, if we've got a system where we're doing that whole genome sequence directly on patient specimens, then we don't need to grow the bug at all. We'll have that information even faster. I don't know how many of you guys know PulseNet. PulseNet was this great idea that the CDC came up with decades ago of doing molecular typing of foodborne isolates. Now they've switched over from pulse field gel electrophoresis to whole genome sequencing. But it's a great thing, if somebody gets a salmonella isolate in Idaho and someone gets a salmonella isolate in Kentucky, they both get sequenced, and the CDC knows that they're both the same isolate. And then you can look and see, what do these people have in common? Do we have an outbreak of salmonella that's hitting 5 different states? And where is it coming from? So it's great. I kind of look at this and say, why not move this on also into hospital epidemiology? If we can do whole genome sequencing on patient specimens, then is there a possibility of doing something like the data gets fed into some massive computer? It's going to have to be a massive computer, but then we can look at it and say, okay, look, this isolate of MRSA is the same one that these people over here got, and we can look at epidemiological links, not just within our hospital, but epidemiological links with other hospitals in our city, in our state, in the whole country. Pipe dream, okay, I know, but I figure as long as I'm kind of making stuff up in this presentation, I might as well go there as well. So, okay, lastly, let's talk about a big topic that everybody's talking about these days. It's a hot thing. When we do something like this, when we take a specimen and we do whole genome sequencing directly on that specimen, uh, we're not just getting the single organism. We're not just getting a few organisms. We're getting the entire population, all the bugs that are in that specimen. So we're basically getting the microbiome of that site. Now, if it's poop, we all talk about stool microbiome, but it could be throat, it could be the genital tract, it can be the urine, it could be the microbiome of a wound specimen, of course. We could do that. So we can talk about the microbiome a lot. There's tons of stuff out there. You go looking for microbiome stuff. It's all over the place. I'm just putting this in here so you have a few references from the last few years. These are review articles that kind of go over a bunch of stuff. And this book here, I threw it in there. I try not to read science stuff when I go home at night because, I want to kind of escape. But I read this book a few years ago. It's it's kind of a good overview. It's kind of exciting to read some of this stuff about what's going on with microbiome stuff. One of the big things that people are really excited about is the relationship between the microbiome, the stool microbiome, and obesity. This is a paper here. It has not been published. This is going to be presented at the European Congress on Obesity in May. These people took a bunch of people and looked at their stool microbiome. So people who are slim had high levels of Christensenella minuta. This is well known. There's been lots of studies which have shown that this particular bug is in high amounts in people who are thin. You can imagine that people are jumping on this. You can imagine, of course, there's companies that are going to be selling probiotics containing Christensenella minuta, and they are. You can probably go to Walgreens and buy some Christensenella minuta. They also looked, overweight males tended to have a lot of these two bugs. Overweight females tended to have a lot of these three bugs. Now, the big issue, of course, is this an unbalanced microbiome that is causing obesity? Or is this obesity that is leading to an unbalanced microbiome? That's not addressed. And the huge majority of papers that are out there about the microbiome do not really address that. But if it is the microbiome that's causing obesity, then you open up the possibility of preventing it or treating it by rebalancing the microbiome. So everyone was really excited. This paper is really old now. This is from 2013, but this paper has been cited over and over and over again. We talk about this paper all the time. They took twin mice. These were from the same litter, a thin mouse and a fat mouse. If you take poop from the thin mouse and transplant it into the thick mouse, not the thick mouse, the overweight mouse, it loses weight. The fecal transplant will actually cause the organism to become slimmer. And it can go the other way as well. You can make this mouse fat by giving him some of his brother's microbiome. You can imagine, I'm just, off the top of my head thinking, we talk about fecal transplants with C. diff. I shouldn't say this. This is rude. I'm sorry. But, it's like if I needed a fecal transplant for C. diff to say, okay, fine, I'll take a fecal transplant, but I want it to be from a thin person. Okay. Now, does it work in humans? Well, there have, of course, been some attempts. This is one that just came out a couple of years ago now, year and a half or so ago. They tried it in patients, in humans, and it did not work. So there's still some work to be done on that. Here's just some things. You can Google the microbiome, you go all over the place. You can find all kinds of studies that show that there's an association between microbiome composition, and this is going to be gut for the most part here, and all kinds of different conditions, behaviors, diseases, all kinds of things. These are associations. So you really don't know what the deal is here. You cannot assume cause and effect with these. There's also some interesting thing out there about mother's vaginal microbiome on a child's health. Even things looking at things about a child's health when they get to be 10, 15 years old are associated with mom's microbiome at the time of birth. So interesting, cool stuff, but we know so little about it at this point. But what that means, if you get back to this, if the microbiome is causing the disease, then we can talk about prevention and treatment. If the disease is causing the microbiome, then we could possibly get some diagnostic help of the disease by looking at the microbiome. So I can imagine 10, 15 years from now, a whole new branch of medicine on microbiome management, whether we have infectious disease doctors who move into this kind of thing, or whether we have a whole new breed of doctors who are microbiomologists or something, looking at restoration of the microbiome, either after antimicrobial treatment or after disease, using the microbiome to prevent disease or to treat diseases. I can see that occurring, which opens up a whole new job for the microlab. Since what we'll be doing is this whole genome sequencing directly on patient specimens, you know, that's pretty cool. We'll be measuring the microbiome anyway. And so the job may not be so much to figure out what one bug is causing this patient's pneumonia or what one bug is causing this infection, but rather let's look at the entire list of bugs that we get out of there and see how that relates to other conditions. Would it be possible to be using the microbiology lab to get information about something like autism or obesity or something? And then monitoring that microbiome to ensure that it's properly balanced, properly restored, or even using it to treat disease. There's a whole lot of stuff going on with this microbiome stuff. It's infancy. It really is early. There's a lot of work that's got to be done first, but it's going to be pretty hot. So if you look at micro now, you know, we do cultures to grow the one bug. This is the one bug that we think is causing this infection. We do phenotypic susceptibility testing on that one bug all by itself in a pure culture. Sure, we can do some targeted sequencing. We can send out abscess specimens or sputum specimens, whatever, to target certain organisms for those bugs that we can't grow. And yeah, we can take an isolate that we have cultured and then do whole genome sequencing to get all kinds of information about it. But 2040, what might be going on is we do whole genome sequencing directly on patient specimens. So we identify pathogens without having to culture them. We get information about that, including how to treat what antibiotics or what other type of treatments we can do based upon the genetic data. We can do epidemiological typing immediately so that we can detect outbreaks or transmissions right off the bat. And we can look at the characterization of the microbiome to say, is this associated with other illnesses or other conditions? And can we treat the microbiome or help restore the microbiome to a proper balance to help out with those situations, help out with those conditions? Like I said, I am no expert in any of this stuff. This is all kind of me just geeking out on it and kind of saying this is going to be kind of cool. I will be gone by 2040, so I'm looking for the next person to come in and actually apply some of this stuff here in our laboratory. But I will be happy to address any questions if I can stop sharing. Okay. Be happy to address anything if anybody wants to shoot me down or to come up with some other cool observations.
Anybody have any questions? I think it's very interesting that I love the idea of being able to look and see epi links across communities and the states. I think that'd be very interesting to your point. So
I think it would be super cool, you know. And this whole idea of doing sequencing on pathogens of importance as soon as you isolate them, you look at that and say, God, that just sounds so expensive and so crazy to do. But there are studies, and of course, there are companies out there that will do this, who will tell you, no, 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 you'll save so much money. You spend an extra, this is what I've always tell people, you spend an extra $1 million on your micro lab, you can save $5 million in other areas of the hospital.
Right.
And there are studies that have shown from an infection control standpoint, you can save a lot of money, save a lot of lives by doing this typing immediately,
Yeah. Yeah,
prospectively.
I could imagine that it would, I mean, it would just help you to figure out even where to, in some scenarios, where to really start. So,
Yeah.
yeah, very interesting. Well, we look forward to 2040.
Hope you all enjoy 2040. I will be sitting in a nice comfy chair somewhere, probably screaming at The Price is Right.
Well, thank you very much for presenting today. And if no one has any other questions, then we will see you all again back on May 15th for our next Grands Rounds. Thank you all.